Imagine a nice winter morning. You're married and recently had a baby. Everything seems to be going well, but then, it's confirmed. He's infected with Respiratory Syncytial Virus. But this is not an isolated occurrence because most children would have been infected by RSV by the age of 2. RSV infections could lead to other complications or more serious diseases such as asthma, pneumonia, bronchiolitis, or recurrent RSV infections. The problem with RSV is that RSV infections create serious problems worldwide. There is a huge global burden. It causes people to fall sick and some may even die. Annually, it causes 64 million infections and 160,000 deaths. Now, let's have a look at some of the risk factors for RSV infection in a population. Those at risk of being infected include infants, particularly those who are born prematurely or those who are born with a low body weight. Other than the young, it also affects the elderly, who are adults above the age of 65. Typically, adults with a weak immune system or who are suffering from other illnesses tend to have higher risk of infection. Also, males have a greater risk of infection than females. Other factors including smoke exposure, heart and lung diseases, and also the climate, especially during winter and spring, also affect the risk of infection. Now that we have established that RSV infections are serious and that it is a global burden that needs to be addressed, you might be thinking, oh no, what can we do about this problem? What options do we have? Well, this diagram represents how a cell is infected. Typically, we would want to either block the virus from binding to the cell, or block the fusion of the virus in the cell, or prevent the virus from making more copies of itself inside the cell, which we usually refer to as viral replication. There has been a lot more research into how RSV can be prevented and treated, and common options include vaccines, monoclonal antibodies, and drugs. Vaccines. They prepare your body to fight against an infection, like how an army prepares for war by training up more troops before they even encounter the enemy. Monoclonal antibodies. They bind to the virus and prevent it from contacting or stimulating the cells. This could be similar to how your sibling holds onto the TV remote to prevent you from changing the channel. Drugs, which may function in many different ways, including those mentioned previously. Recently, various studies into the immune system have revealed the potential of antimicrobial peptides, and as the name suggests, they are proteins that work against microbes. These include cathelicidins such as LR37 as potential antiviral molecules. LR37 has demonstrated activity against a range of viruses such as vaccinia virus, varicella zoster virus, influenza A virus, human immunodeficiency virus, herpes simplex virus, and dengue virus, and most importantly, respiratory syncytial virus. Specifically, LR37 was shown to exhibit antiviral activity against RSV in vitro, and the paper that published this finding found that LR37 reduced the spread of infection, decreased virus production, limited the cell death caused by infection, provided short-lived protection against subsequent RSV infection, and also that LR37 could interact directly with RSV. Hence, I flew 6,787 miles from Singapore all the way to Edinburgh to join the Davidson group. This is so that I can study LR37 in greater detail. And here's my project title, Examining the Potential of Antimicrobial Peptides as Novel Antivirals Against RSV. I know that sounds like a lot, but hang in there, I'll explain it. I want to understand how LR37 is antiviral towards RSV and how it changes our body's immune response to RSV. In simpler terms, I just want to know how LR37 fights against RSV, just like two boxers in a boxing ring. This is so that I can gain insight on whether LR37 can work as a cure or a preventative measure for RSV. Generally, my hypothesis is that LR37 is antiviral towards RSV. And how do I plan to test this hypothesis? Firstly, I want to see how different conditions affect RSV infection. Secondly, I want to characterize how LR37 affects the spread of RSV infection. And thirdly, I want to explore the effect of different inducers of CAMP on RSV infection. To study the first aim, I will add RSV and LR37 in different sequences. Essentially, this is like making breakfast. What happens if I add the milk and then the cereal? Or what happens if I change this up? Next, I will study how LR37 affects the spread of infection over a period of 72 hours. Essentially, this is like if I left a sweet on the ground and wondered how many ants it will attract after a period of, say, 24 hours, 48 hours, or even 72 hours. Lastly, I will add different chemicals to the cells to see if the cells will make more LR37. Essentially, this is like cooking, where we often consider if different sauces or spices are able to make the dish tastier. I will perform all these experiments so that hopefully there can be a future where RSV infections can successfully be prevented or treated, and young families everywhere can continue to enjoy nice winter mornings with one less worry on their minds. Thank you so much for your kind attention, and I would like to give special thanks to the Davidson Group at the Queen's Medical Research Institute of the University of Edinburgh.